Hi, welcome to the Sybil Cluster Report Page screencast. This screencast will cover the features of the Sybil Cluster Report page. The protein cluster page can be accessed from a variety of places, but for this demo, we'll get there through the cluster search page. From the strep-pneumo homepage at strep-pneumo-sybil.igs.umaryland.edu, we'll click on the Protein Cluster Search button. Then, we'll click on the Search for Clusters tab. Let's search for clusters that contain the gene product name protease. Let's find out a bit more about this serine protease gene cluster. We'll click on its name to bring up the cluster's report page. The cluster summary section shows the name and a description of the clustering used, the number of proteins in the cluster, the average percent identity and coverage of the pairwise hits, and two buttons to download the DNA or protein sequences of the genes in the cluster. The clustered proteins section provides a list of the cluster members and serves as a form for drawing genomic context and BLAST-P images, as well as remaking the multiple sequence alignment found at the bottom of the page. The order column contains a drop-down that allows vertical ordering of the genes in the subsequent panels. I'll demo this a bit later. The organism column gives the source organism name. The protein and alternate protein name give two potentially different display names for the gene. The name listed in the protein column is the name that has been set up as the display name in the remaining images. The length of the gene, number of exons, and gene product name round out the columns of this table. This table can be hidden by clicking the Hide button on the left side of the table header. The Load Data table gives a single button to redraw all the remaining panels. Let's click that button now. The genomic context image contains a visual representation of the cluster members selected in the clustered proteins table above. Genes that cluster together are connected by pink or gray polygons. Links between genes in the cluster we're exploring now appear pink, while neighboring cluster links appear gray. Clicking on a cluster yields a pop-up that contains a link to the cluster's report page, several links to the genomic comparative view, and a list of the genes in the cluster, both genes in the current display and those not currently displayed. The BLAST-P results area gives a graphical representation of the best BLAST hits from all the members selected in the clustered proteins table above. Finally, the cluster alignment area shows the multiple sequence alignment of the genes selected in the clustered proteins area. This alignment can be regenerated using nucleotides if desired. Let's return now to the clustered proteins table and show how we can select just a subset of the cluster members. I'll start by choosing Deselect All at the bottom of the table. Now I can click the word Add in the first column to add a gene. Clicking Remove removes a gene. Clicking the button Select Inverse will deselect all genes currently selected and select all genes that are currently not selected. Once the desired members are selected, we can go back and redraw one or all of the sections we just saw. In this case, I'll just click Redraw All Panels again. The same options appear in the genomic context image area as appear in the protein report page's context image area. We can choose a new pad, whether or not to show gene labels, and what additional feature types we'd like to see. We can also select an image width in pixels, as well as request a downloadable SVG image. I'm going to go ahead and select all the feature types, display the gene labels, and set a 10 KB pad, and we'll make the image 1200 pixels wide. As we scroll further down the page, we can see that all the panels have been reloaded to include only the genes we've selected above. Likewise, the genes have been reordered according to the ordering we specified in the drop-down boxes.